Hi everyone, this is Anafal Balushi and Noor Salhi, and today we are presenting our honors project, which is demineralization of dairy whey product using MCDI. First, let me define what is whey. Whey is a byproduct from cheese, yogurt, and casein production. It's a yellow-colored, thin, and watery liquid. The high mineral content in whey product has limited its usage in various applications due to its salty taste. Therefore, demineralization is crucial for human consumption because it reduces the level of mineral present in whey. Our objective is to investigate the efficiency of MCDI technology as an alternative to current whey demineralization technologies to add value to both the dairy industry, and also we want to contribute to knowledge in the MCDI domain.
Now let me introduce you to membrane capacitive deionization, which is an innovative and cost-effective technology that separates dissolved ionic species by electroadsorption process. As shown in the poster, the MCDI cell consists of two acrylic plates, graphite sheets, a gasket sheet, activated carbon electrodes with either an anion or cation exchange membrane attached to it, and a nylon cloth spacer channel. The methodology undertaken to perform this study is clearly illustrated in the poster. Step 1 is electrode assembly. Step 2 is electrode performance testing, firstly for a simple NaCl solution to test the capacity of salt removal. Following that, the electrode performance was testing when passing whey solution through the cell. Whey model synthesis and the experimental setup will be demonstrated in the shortcoming video. To access the video, please scan the QR code shown in the icon.
Start by preparing the model whey solution containing 1500 ppm of NaCl. We mix 3.75 grams of whey protein isolate with 150 ml of deionized water in a beaker using a magnetic stirrer at 1.5 rpm and at room temperature until it reaches equilibrium. Then we add 0.23 grams of NaCl to the whey solution, and then the solution is mixed.
Moving on to the experimental setup, the MCDI experiments were carried out in both batch and single pass operation modes. The operation mode shown in the video is a batch mode, at which the system consists primarily of a beaker magnetic stirrer plate, the MCDI cell, a DC power supply unit, a pressure sensor, and a conductivity probe. At this operation mode, the conductivity probe measures the variation in the recycling reservoir, which was done by connecting the conductivity probe to Vernier graphical analysis for software. The pressure change within the MCDI unit over time is measured using a pressure sensor connected to Arduino software. Before further proceeding, it was ensured that no air bubbles were present in the inlet pipings. The purify step was carried out by applying the electric voltage using a DC power module. During the desorption step, reversed current mode was used, at which the polarity of the electrode cell is reversed, causing the collected ions to be pushed back into the spacer channel.
Step 3 is electrode performance characterization. The basis for scale-up calculation of an MCDI cell relies on the parameter that will be obtained from fitting the modified Donan electric double-layer model to data obtained from experimental study. Experimental run with a simple NaCl yielded a VMI of 0.17 ml. And experimental run with whey protein isolate solution yielded an effective VMI of 0.1 ml and an average salt adsorption ratio of 0.04 mg per second per gram of carbon. As you can see in the graph, the principle of electrosorption on whey solution containing mineral work almost as efficient as NaCl only solution. Inductivity coupled plasma was performed for the initial whey solution and the demineralized whey solution to evaluate the reduction in salt removal. As shown in the table, the result illustrates that there is no ion selectivity in the current design, which is mainly because the initial whey solution had a comparable concentration of these cations, which had led to an equivalent percentage of ion removal.
Falling was observed within the MCDI unit since the pressure was increasing over time. It was determined that the falling did not affect the electrode salt performance. Thus, it was concluded that the falling occurred mainly on the spacer channel, not on the carbon electrode. To study the possibility of reducing the hydraulic resistance within the MCDI unit, the MCDI cell was redesigned where a double spacer channel was used instead of single one. The results showed that the pressure change within the MCDI unit was lower when using a double spacer channel. Different cleaning agents, including alkaline and acidic cleaning agents, were used to remove the filled material. The results showed that acidic cleaning did not effectively remove the fold species. However, the fold layer transformed into a gel-like material after alkaline cleaning. This indicated that the falling layer consists mostly of organic foulant.
The team concluded that MCDI is capable of reducing 25% of the salt content in Whey solution. Although this is lower than the demineralization efficiency achieved by existing technologies, the capex of a large-scale MCDI plant used to demineralize Whey is about half the cost of existing technologies. Now, further research is required for investigating cation selectivity in a more complicated multi-ion system, such as using acid or sweet Whey.